Hello and hi to Madam Ainol and Dr. Wan. My name is Max Michael Sampson, and today I will present my individual project for molecular techniques and tools, which is immunoprecipitation. This is the content overview. There are 4 subtopics of content, which are the introduction, the information content, conclusion, and references. For the information content, I have subdivided it into 3 parts, which is part 1 is the molecular basis technique, which is how the IP works. Part 2 is the laboratory tools or machines that involve on how to do the IP. And part 3 is the example of research that has been carried out using IP technique. So what is IP? IP, or immunoprecipitation, is a technique of precipitating out a protein antigen out of a solution using an antibody that specifically binds to a particular protein. This means that in order to precipitate out the desired protein or protein of interest out of a solution that may contain a lot of other proteins, organelles, or other molecules, we can actually use antibody that has the ability to target or to bind to that specific protein and precipitate it out. So there are 3 general ways for the technique to be done, which is firstly by incubating the sample with antibody against the protein of interest. Second is to separate the antibody or protein complex from the remaining sample, and then we can analyze the protein. And there are 5 types of immunoprecipitation technique that are known, which are the individual protein immunoprecipitation IP, co-immunoprecipitation co-IP, chromatin immunoprecipitation chip, ribonucleic protein immunoprecipitation RIP, and tagged protein immunoprecipitation. But today I will be going to focus more into the general one, which is the individual protein immunoprecipitation, or short form IP. And this technique requires the use of an antibody that is specific for a known protein. So meaning to say that this antibody have already been exposed to a known protein so that it can isolate the same protein from a solution that contain the particular protein. Moving on to the molecular basis of immunoprecipitation technique, or how the IP works. In the first diagram of microcentrifuge tube, we can see that the lysed cell extract contains 2 types of different protein. The goal here is to precipitate out the protein of interest. To do so, we can use antibody that has been coupled with agarose or magnetic beads. In the second diagram, in the extract, we can see that the antibody has successfully binded to the protein of interest. Upon centrifugation, the beads will somehow bring the protein to pellet out at the bottom of the tube, while the supernatant will be discarded. Eventually, this process will be repeated several times until we get the eluate protein. Only then, we are able to analyze the protein of interest. So, what are the equipments and tools that are required to do the IP? So, the equipments that are needed are microcentrifuge tubes, microcentrifuge, ice bucket incubation, gel electrophoresis kit, reagents like cell lysis buffer, SDS sample buffer, SDS running buffer, transfer buffer, phosphate buffer, saline, and of course the fundamental elements which required protein AG, antibody, and agarose or magnetic beads. There are several aspects that need to be considered when choosing between agarose beads or magnetic beads. Agarose beads has a larger surface area compared to magnetic beads. Thus, agarose beads has a greater binding capacity compared to magnetic beads. On the other hand, magnetic beads has a faster diffusion rate compared to agarose beads since it has a smaller surface area. Agarose beads also tends to have an extensive wash which may lose too many samples, while magnetic beads require less washing, hence more sample can be obtained. Next, we are going to talk about the IP protocol, which involves the preparation of antibody protein AG agarose beads, preparation of cell extract, and immunoprecipitation process. So we are going to talk about the first protocol. Please note that the protein AG is actually a protein antigen that helps the antibody to mark and target the protein of interest. So firstly, the protein antigen agarose beads will be washed with cell lysis buffer, and then it will be go into the microcentrifuge and spun for 2 minutes, and then the supernatant will be discarded, and the beads will be washed again 3 times with the same cell lysis buffer. Then, a diluted antibody will be transferred into each of the agarose beads sample. This mixture of antibody protein antigen agarose mix will be shaken and rotated at 4 degrees Celsius about an hour. Lastly, the protein antigen will be spun about 2 minutes to detach the antibody beads, and the beads will be washed again 3 times with the same cell lysis buffer. Next protocol is the preparation of cell extract. Firstly, the sample cells are collected and centrifuged for 5 minutes at 4 degrees Celsius. Next, the supernatant will be discarded and immediately add ice cold lysis buffer to the cells and vortex it again. Then immediately incubate it for 30 minutes in ice bucket. To release the proteins from the cells, you can either freeze store the samples with dry ice twice or sonicate it in a pre-cooled. Lastly, the lysate will be spun centrifuge for 10 minutes and the supernatant will be kept. The third and last protocol is the immunoprecipitation process. The protein concentration from the supernatant that is kept from the second protocol will be taken with lysis buffer. Then, the cell extract will be mixed with antibody protein antigen agarose that has been made from the first protocol, and then rotate it for about 2 hours, and the agarose beads will be collected after microcentrifuge. The supernatant will be discarded, and the beads will be washed 3 times with ice cold cell lysis buffer. After the final wash, the supernatant will be removed and SDS sample buffer will be added. Boil it for 5 minutes at 95 degrees Celsius and spin the beads at maximum speed for 5 minutes at room temperature. Then the supernatant will be pipetted out. The sample will be loaded into a thick gel, and then it will be run in the gel electrophoresis by immunoblotting using Western blotting technique. Moving on to the research example that I came across from Science Direct, which is about Alzheimer's disease, and the journal title is A Simplified and Sensitive Immunoprecipitation Mass Spectrometry Protocol for the Analysis of Amyloid Beta Peptides in Brain Tissue. So it is about the analysis of peptides, which requires immunoprecipitation method to precipitate out proteins. But instead of using agarose beads, they use magnetic dynabeads, and the dynabeads is pelleted out by using magnet. Otherwise, the protocol is much like as the protocol that I have been mentioned before, but the eluate at the end of the research was dried out so that it can be used for the mass spectrometry analysis. Hence, to conclude, immunoprecipitation is indeed the simplest form of extracting protein when compared to the other types of immunoprecipitation techniques. However, this Technique has a limitation of targeting other or multiple proteins since the antibody can only detect that particular protein after it has been exposed to the specific protein antigen. Third, different type of bits used in IP will surely impact the result differently. And lastly, IP can only target non-proteins. And these are my references for this presentation. Thank you.